Hi everyone, it's Mrs. Bustamante. Today in this video, we're going to talk about the discovery of DNA structure and replication a little bit. Really, historically, how did we get to the point that we are now? How do we know the things that we do about DNA now? What experiments and individuals played a key role? What did they discover? How did they discover it? And why is it important? And then if you see a star next to somebody, that means that's somebody you should really take a little bit of time and importance to know. You might see them again. So when we think back, right, where did we leave off? Kind of with Mendel, right? This idea that we could look at all these different plants and we could see traits pass on from one individual to the next, but we still don't know exactly what is causing that, right? So that kind of brings us to this idea of somebody named Friedrich Michener, and he discovers a new compound that he calls nuclein. It's in the cell of white blood cells. Later, we would go on to call this DNA, right? But we don't know this at the time. And he just discovers that this particular compound has lots of nitrogen and phosphorus in it, which if we think about what we know about DNA now, right, makes sense. We're starting to discover more about the pieces that might play a role. We need that compound in order later on we know that it's that. The person I really want to talk about here, though, is Frederick Griffith. Frederick Griffith was a scientist. He was actually studying pneumonia with mice in order to develop a vaccine against it, specifically Streptococcus pneumonia, which is caused by a bacteria. So he has these two strains of bacteria, and he injects them both in mice in different ways. One of those strains is virulent, meaning it's going to cause the mouse to get sick and die, and one of them is non-virulent, which means non-pathogenic, that mouse lives a normal, healthy life, okay? So what do we see? When he ingests the smooth strain into the mouse dies, as expected, right? When he puts the rough strain into the mouse lives, okay? So now we think smooth kills, rough lives, no problem. Then he goes ahead and he heat kills the smooth strain of bacteria that would typically cause pneumonia. He injects it in the mouse, and what does he notice? Well, that mouse lives, okay? So somehow something he's done has basically killed the part of the bacteria that is causing the mouse to get sick and die, right? By this part of the experiment, okay? So he goes on and does one more thing. He takes the rough strain that was heat killed and the smooth strain, and he puts it into the mouse. And what does he see? Here he sees that the mouse actually dies, which is interesting, right? Because we took two strains, both of which individually did not cause the mouse to die, and we inject it into the mouse. Wonder what's going to happen, right? Well, what do we notice? Because the mouse died, somehow the living R bacteria has been transformed by an unknown heritable substance from the dead S cells. So what this really did is it allowed those R cells to make capsules that then caused the mouse to die, right? Caused the mouse to get pneumonia and die. Later on, we would call this process transformation. It's a change in phenotype and genotype due to the assimilation or the insertion, accumulation, basically putting it in to the genome of external DNA by a cell. Later, it would be determined specifically that this information is actually DNA, but we're noticing here from Griffith's experiment really that there is some substance in cells that gets passed along and really can change other cells in order to replicate and move on, right? So there is some sort of molecule that is being inherited and can cause changes in one cell to the next. So next we have Alfred Hershey and Martha Chase. We call this the Hershey-Chase experiment. And they actually are studying how viruses can infect bacteria. So let's take a moment to talk about what are viruses. Viruses are much simpler than cells. They're really essentially some DNA or RNA inside of a protective protein coat some protective capsule, and then they have some mechanism for basically injecting that into the cell because they cannot reproduce and make more viruses without some sort of a host. So they have this mechanism that they can inject it in the cell, and basically they take over the cell's metabolic machinery so that it can replicate and make more. So here's what Hershey and Chase do. They're going to label the bacteriophage, which sometimes we just call a phage, which is a virus that basically infects a bacteria. And they do, some of them they label with radioactive sulfur, and some of them they label with radioactive phosphorus. And we're going to talk about what happens to that in a second. So some things to think about here, right? DNA only contains phosphorus. It does not contain sulfur. So when we label something with radioactive phosphorus, right, it would probably show up in the DNA because it wouldn't tag the DNA if we add radioactive sulfur, right? Proteins, on the other hand, only contain sulfur. They do not really contain phosphorus. So when we're tagging our bacteriophages or our phages, right, it's going to tag the protein coat with sulfur, and then the DNA inside would get tagged with the phosphorus, right? So looking at what is present when would tell us really what part of a phage is infecting cells and transmitting on and replicating more. So here's their experiment. Here's kind of what they do. Okay, they do 2 batches, right? In their first batch here, which is our pink one that you can see, the phages were grown in radioactive sulfur. And remember, sulfur is going to basically tag the protein coat. So they kind of like blend them up essentially together, agitate the mixture so that those phages infect, if you will, the cell of bacteria. Then what do? They centrifuge it. What does centrifuging do? Really spins something really, really fast, so it separates it into the layers. What do they see in these layers, right? In these layers, they're going to measure the radioactivity in the pellet and in the liquid. Okay, so when they do this, we're looking at where is our radioactivity, essentially in the protein coat, right? And they found that it was in the liquid, the top section of the centrifuge stuff. And then this down lower part was your pellet. That was your bacteria cells and its content, okay? So we did not tag the part that essentially has DNA on it. This was the protein, didn't move on to the bacteria cells, right? In batch 2, the phages were grown in radioactive phosphorus, which was incorporated into the phage DNA. Again, we blend it together, mix it into the cells, centrifuge it out. So we get the cell parts at the bottom because they're heavier and the lighter liquid at top. Well, what do we notice? We notice that there is radioactivity of the phage of the DNA in these pellets down here, right? So what is that telling us? That tells them that phage DNA has entered the bacteria cells, but not phage proteins, okay? So radioactive phage DNA was released into new phages, or was able to make new phages that also were radioactive. So that shows that DNA and not the protein is actually where the genetic information is and how it gets passed on from one generation to the next, because we see it labeled with that radioactivity in the cell parts, and we do not see that in the protein part. So that's Hershey Chase, and they really discovered that DNA is the heritable information through this experiment. Some other important players, Kossel, he described nitrogen bases, basically that there are purines and pyrimidines, right? And they're hydrophobic. So when we think about now that we know DNA is a double helix, we know that they're hydrophobic. So it makes sense that they're on the inside of your double helix because they don't like water, right? Later on that becomes important, though he doesn't know that right now. Levine finds that DNA is made out of units, a sugar or phosphate base. He calls these nucleotides, right? Later on we call these monomers, and we know that they are the building block. Again, this sugar phosphate is hydrophobic. So now when we think about the structure of DNA, we want hydrophilic, those sugar phosphate bonds on the outside in watery like substances make sense with those bases on the inside, more protected. Linus Pauling, he's actually from Portland, Oregon, and he uses techniques to basically make it so we can visualize and better understand molecular biology. And his techniques get used later on by Watson and Crick to really help us understand DNA better. We have Erwin Chargaff. What's his contribution? Really, he says by looking at lots of DNA samples, the amount of C always equals the amount of G, and the amount of A always equals the amount of T. Doesn't matter what organism you're looking at, they always seem to be equal in percentages. Maybe the percentages differ a little bit between organisms, but A and T and G and C are always equal. So this is really our base pair rule that we know and use now. Okay, Maurice Wilkins. We should probably put a star, I guess, by Chargaff. His rule comes up a lot. It's kind of important to understand him too. Now we get to Maurice Wilkins. Okay, Maurice Wilkins actually later will receive the Nobel Prize for his work in the discovery of the DNA structure, but he discovered that DNA has a crystalline structure. He used X-ray crystallography to make diffraction patterns, basically showing how molecules were put together. Different molecules, different patterns will show differently on these pictures. Now, Rosalind Franklin also worked in the same lab as him, and there's some stories about how they didn't get along and how Wilkins thought Franklin was hired to work for him, and she doesn't really think that, right? And over time, it's kind of come out that she was really the one who was the X-ray crystallography expert. We're going to talk about Watson and Crick too, and she's the one who kind of had the chemistry background to do it. She's really famous now because she took what we call Photo 51. Photo 51 is a clear diffraction image of DNA, and it shows that the structure of DNA is actually something called a double helix, which we know now. Here is her picture, Photo 51. And this banding pattern is what tells us it is that double helical structure. Now, why is this a little bit controversial, right? Well, we typically, most of us, know Watson and Crick as being the ones who discovered the structure of DNA. Well, kind of what happens is Wilkins sort of takes Rosalind Franklin's photo, Photo 51, and shows it to Watson and Crick, who are having a hard time discovering exactly what the structure of DNA looked like, right? And so it's with this Photo 51 that Watson and Crick actually say, oh, the structure of DNA is a double helix. And they published this paper in 1953 about the structure of DNA, and it is the double helix in this scientific paper called, or organization called Nature. They actually also published Franklin's paper because all of this kind of happens at the same time, but they put it after Watson and Crick, so it kind of looks like that her data really just supports what Watson and Crick discovered. So kind of they stole her data and used it to write a paper and figure it out without her consent. In 1962, Wilkins, who worked with Rosalind Franklin, Watson and Crick all received the Nobel Prize for their work in DNA, but Franklin did not. She died in 1958 of cancer, probably due to the work with her radiation, X-rays, right? And you have to be alive to receive the Nobel Prize, so she was never given the Nobel Prize, although it's suspected that she probably should have if she was alive.